Welcome to B002. In our last lesson on cell theory, we learned that all life is made of cells, and we saw the big picture difference between prokaryotic and eukaryotic cells. Now we are going to go much deeper. We will look at the internal architecture of each cell type in detail, every major structure, what it does, and why it matters. This topic is one of the highest yield areas in NEET biology. Multiple comparison questions appear every single year. Let us build a thorough understanding. Let me give you a mental model that makes the difference between prokaryotes and eukaryotes immediately clear. Think of two cities. The first is an ancient settlement, no walls, no separate districts, the market and the homes and the workshops all share the same open ground. Simple, efficient, but limited in complexity. The second city is modern, it has a secured government district where the most sensitive decisions are made, separate industrial zones, a dedicated power station, a waste processing facility. Both are cities. Both function. But the modern city can coordinate far more complex activities simultaneously because different functions are separated into different spaces. Prokaryotic cells are like the ancient city. Everything happens in one open compartment. Eukaryotic cells are like the modern city. They have a nucleus that secures the DNA, mitochondria that generate power, an endoplasmic reticulum for manufacturing, a Golgi apparatus for distribution, and lysosomes for waste management. This compartmentalisation is what allowed eukaryotic cells to evolve the complexity needed for multicellular life. Let us go through the key structures of a prokaryotic cell one by one. The cell wall in bacteria is made of peptidoglycan, a mesh-like polymer that gives the cell its rigid shape and protects it from bursting due to osmotic pressure. This is actually the target of penicillin, which kills bacteria by disrupting peptidoglycan synthesis. The plasma membrane is a phospholipid bilayer present in all cells. In prokaryotes, it is the outermost functional barrier since there is no nuclear membrane or organelle membranes. The nucleoid region is where the DNA is located. It is circular, double-stranded DNA, but it is not enclosed in a membrane. This is the key distinction. Prokaryotes also have plasmids, small circular pieces of extra DNA that are not part of the main chromosome. They carry genes for things like antibiotic resistance, which is why bacteria evolve resistance so quickly. The ribosomes in prokaryotes are 70S, made of 30S and 50S subunits. This matters because our antibiotic drugs are designed to target 70S ribosomes without harming our own 80S ribosomes. Flagella allow movement, and pili are hair-like projections used for attachment and for transferring genetic material between bacteria through a process called conjugation. Now let us explore the eukaryotic cell. We will cover the nucleus and the endomembrane system on this slide. The nucleus is enclosed by a double membrane called the nuclear envelope, which has nuclear pores that allow selective transport of molecules in and out. Inside is the nucleolus, which is not a separate organelle but a dense region where ribosomal RNA is assembled. The DNA in eukaryotes is linear and wrapped around proteins called histones. This is fundamentally different from prokaryotic DNA. The rough endoplasmic reticulum is studded with ribosomes and is the main site for synthesizing proteins that will be secreted out of the cell or embedded in membranes. The smooth endoplasmic reticulum has no ribosomes. It synthesizes lipids including phospholipids and steroid hormones. It also detoxifies drugs, which is why liver cells, which do a lot of detoxification, have an abundance of smooth ER. The Golgi apparatus acts like the cell's post office. It receives proteins from the rough ER, modifies them, for example by adding sugar chains, then packages them into vesicles and sends them to their destination, either within the cell or for secretion. Lysosomes are membrane-bound sacs full of digestive enzymes. They break down old organelles, cellular debris, and pathogens engulfed by the cell. Now we come to the two most fascinating organelles in eukaryotic cells, the mitochondria and the chloroplast. Mitochondria are called the powerhouses of the cell because they produce ATP through cellular respiration. They have two membranes. The outer membrane is smooth. The inner membrane is folded into structures called cristae, and these folds dramatically increase the surface area where ATP is made. The space inside is called the matrix, which is where the Krebs cycle reactions happen. Now here is something remarkable: mitochondria have their own circular DNA and their own 70S ribosomes, just like prokaryotes. This is strong evidence for the endosymbiotic theory, which proposes that mitochondria were once free-living bacteria that were engulfed by a larger cell and gradually became permanent residents. Chloroplasts are found only in plant cells and algae. They also have double membranes, their own circular DNA, and 70S ribosomes. Inside, they have thylakoid membranes stacked into structures called grana. This is where light reactions happen. The surrounding fluid, the stroma, is where the Calvin cycle runs. The 70S ribosomes in both mitochondria and chloroplasts is the most tested NET fact in this entire chapter. Do not forget it. Here is the complete comparison table that you should be able to reproduce from memory for NET. The nucleus, absent in prokaryotes, DNA sits in an open nucleoid region, present in membrane-bound in eukaryotes. DNA, circular with no histones in prokaryotes, linear with histones in eukaryotes. Ribosomes, 70S in prokaryotes, made of 30S and 50S subunits, 80S in eukaryotes, made of 40S and 60S subunits. Cell wall, peptidoglycan in bacteria, cellulose in plant cells, absent in animal cells. Membrane-bound organelles including mitochondria, chloroplasts, Golgi apparatus, and endoplasmic reticulum are all absent in prokaryotes and present in eukaryotes. Size: prokaryotes are 1 to 10 micrometers, eukaryotes are 10 to 100 micrometers. Study this table until you can fill in every cell without looking. NET frequently gives you one or two features and asks you to identify the cell type. Here are the most important exam tips for this topic. The single biggest trap in this chapter is the ribosome question. Many students memorize the rule: prokaryotes have 70S, eukaryotes have 80S. Then NET asks, what is the ribosome size in mitochondria? The answer is 70S, because mitochondria and chloroplasts evolved from prokaryotic ancestors and retained their 70S ribosomes. Students who learn the rule as an absolute fall into this trap. The most frequently tested comparisons are ribosome sizes, presence of nuclear membrane, DNA structure, and cell wall composition. Be ready to apply these in both directions. Given a feature, identify the cell type. Given a cell type, describe the feature. Viruses deserve a special mention. They are not cells at all. They have no ribosomes, no membrane, no cytoplasm. NET sometimes includes viruses in comparison questions specifically to test whether you know they are acellular. And finally, mycoplasma is a bacterium, a prokaryote, that has no cell wall. It is the smallest known free-living organism. This exception appears in NET questions more often than you might expect. Excellent. Let us close with a summary. Prokaryotic cells have no nucleus, their DNA is circular, sits in a nucleoid region, has no histones, and the ribosomes are 70S. The cell wall is peptidoglycan. No membrane-bound organelles at all. Eukaryotic cells have a membrane-bound nucleus with linear DNA wrapped around histones. Ribosomes are 80S. They have a full set of organelles including mitochondria, chloroplasts in plants, endoplasmic reticulum, Golgi apparatus, and lysosomes. Mitochondria and chloroplasts are special. They have their own circular DNA in 70S ribosomes, which is evidence they were once free-living prokaryotes. The endomembrane system, rough ER, smooth ER, Golgi, works as a coordinated protein manufacturing and shipping pipeline. 2 NET exceptions to remember: mycoplasma is a prokaryote with no cell wall, and viruses are neither prokaryotic nor eukaryotic, they are acellular entirely. In the next lesson, B003, we look at the cell membrane in detail and all the ways substances move in and out of cells. See you there.